Hello, today we are going to discuss two of the four stages of cellular respiration. Cellular respiration is a very important process that results in the production of energy. This energy is then used to fuel all the stages of life. Energy in our bodies is produced in the form of adenosine triphosphate, also known as ATP. Throughout the whole process of cellular respiration, 38 ATP molecules are produced. Like I said earlier, cellular respiration has four stages. The first is glycolysis, which is a 10-step process that occurs in the cytoplasm of the cell. The second stage is the formation of acetyl-CoA. That occurs in the mitochondria. This is where our tutorial will begin. The third stage is the citric acid cycle. This cycle can also be referenced as the tricarboxylic acid cycle or the Krebs cycle. Like the formation of acetyl-CoA, it also occurs in the mitochondria. The final stage of cellular respiration is the electron transport change. This stage produces the most ATP molecules, however, it will not be discussed in the video. Glycolysis is the process that splits a 6-carbon glucose molecule into two 3-carbon molecules called pyruvate. These pyruvate molecules then begin the process of forming acetyl-CoA. The enzyme that helps to facilitate this process is the pyruvate dehydrogenase complex. Pyruvate loses a carbon and it exits the body as carbon dioxide when we exhale. The molecule left behind is a 2-carbon acetyl-CoA. During this process, an NAD high-energy molecule is also reduced to form NADH. This molecule will act as a shuttle for electrons during cellular respiration. Now we begin the steps of the citric acid cycle. The citric acid cycle begins by fusing acetyl-CoA with a 4-carbon molecule backbone called oxaloacetate. By binding these together, we form a 6-carbon molecule called citrate. The process is facilitated by an enzyme referred to as citrate synthase, which makes sense because we are literally building citrate. From citrate, we use the enzyme citrate aconitase to rearrange the molecule to form isocitrate. Isocitrate still contains a 6-carbon backbone. From there, the enzyme isocitrate dehydrogenase cleaves a carbon atom from the backbone of isocitrate and excretes it in the form of CO2. During this process, NAD is also reduced to form NADH. After all these byproducts, we finally get a 5-carbon backbone molecule known as alpha-ketoglutarate. An alpha-ketoglutarate dehydrogenase enzyme then cleaves a carbon off the alpha-ketoglutarate . This is similar to the previous step. As this occurs, another NAD molecule is reduced to NADH. The resulting 4-carbon backbone molecule that is formed is succinyl coenzyme A. After the formation of succinyl-CoA, the succinyl-CoA synthetase enzyme produces succinate, which is still a 4-carbon backbone molecule. During this process, a molecule known as guanosine triphosphate, or GTP, is formed. This molecule is also a molecule used for energy that is similar to ATP. Now that we have succinate, the enzyme succinate dehydrogenase produces fumarate. During this process, a molecule known as FAD is reduced to form FADH2. This molecule has a similar function to NADH, as it is used as an electron shuttle. After fumarate is formed, the enzyme fumarase helps to convert fumarate to malate. Both fumarate and malate are molecules with a 4-carbon backbone. Following the production of malate, malate dehydrogenase enzyme helps to reduce NAD to NADH, all while producing oxaloacetate. Sound familiar? Oxaloacetate is what we use to combine with acetyl-CoA to start the cycle. This is why it is called a citric acid cycle, because it can continue. For each molecule of glucose, the cycle is performed twice. Following this process, cellular respiration continues through the electron transport chain. Now that you've learned the steps of the citric acid cycle, it's time to celebrate. I hope this video helped you learn a little bit more about the processes, enzymes, and carbon backbones of the citric acid cycle. Thanks for watching.